So we just finished up talking, or kind of describing natural selection, and this example is fabulous at showing exactly how it works. So what we see is we have two pictures. The one on the top is a picture of a mantid insect from Malaysia, and on the bottom we have one from Africa. But you can see how different they appear from one another, but how well they fit into their particular niche, right? So the mantid insect on top blends in perfectly with its background, with its habitat, as does the insect on the bottom. But think about what would happen if I picked up one from Malaysia and stuck it in Africa, and then I stuck the mantid insect from Africa on that plant that was in Malaysia. Well, they would stick out like a sore thumb. They would be seen quickly by predators, so probably would get picked off. But what's happened is that over time, as they've moved into new environments, those individuals that have the traits that allow them to survive are going to be the individuals that continue breeding and continue passing on those traits. So, if we're talking about evolution, it's always really interesting to point out this thing called artificial selection. And this is something that's come about because of humans. So humans have the capacity to kind of selectively breed certain organisms in such a way that it might benefit us. So it says humans have modified species over many generations by selecting and breeding individuals that possess desired traits. Okay, so when we select an organism and breed it to produce traits that are beneficial to us, we call that artificial selection. And on the bottom it says, as a result, crop plants and animals bred as livestock or pets often bear little resemblance to their wild ancestors. So I always like to kind of think of if you guys have a certain dog breed you like, or lots of times we're always seeing these new kind of designer dog breeds or designer cats or something like that. Well, we have artificially selected certain traits that we deem desirable, and we've bred those animals in such a way that we can produce them to our benefit. So this is an example of how we've done that. And this is commonly done with different types of crops. But what we see here is originally we had this wild mustard plant. But by selectively breeding certain plants with other plants, we were able to produce all these varieties of food from this one individual plant. So we get kohlrabi and kale and Brussels sprouts and cabbage and cauliflower and broccoli because we have artificially selected for certain traits to be continuously bred until we reached, a new species, if you will, that we are happy with. And Darwin kind of saw our ability to do this. He saw how quickly this could happen. So he came to the conclusion that, hey, if we can selectively breed traits into organisms or into plants quickly, then it shouldn't be too out of the question that this modification or modification of traits couldn't happen in the wild, especially if given hundreds or thousands of generations or even millions of years. So artificial selection can achieve so much change fairly quickly that Darwin reasoned it should be capable of considerable modification of species over hundreds of thousands of generations. Now, another term we need to keep in mind or point out is this term called homology. And homology is pretty cool because you can kind of see common traits that completely different species have in common. So the official definition is similarity resulting from common ancestry. And homologous structures between organisms are basically anatomical resemblances that represent variations on a structural theme that was present in a common ancestor. So in a minute we'll look at examples of different homologous structures that are common between us as well as other species. So here's 4 completely different species that allow us to see different types of homologous structures, and it's really interesting. So on the top it says, even though these species have become adapted for different functions, or even though these traits have become adapted for different functions, the forelimbs of all mammals are constructed from the same basic skeletal elements. And if you look at the pictures, you can see each of these species have a gray bone on the top of their limb. Okay, it may be shaped a little bit differently, but we all have it. If you move on down, you see that this gray bone is going to be connected to two smaller bones. One of them's kind of a peach color, the other one's kind of a pink or red color. From that point, you move down farther and you can see that these two bones are connected to several smaller bones that are illustrated in yellow. And from this point, those small yellow bones are connected to 5 digits, okay, that we share with all these species. So this suggests a common ancestor. Another place where you can see different homologous structures is in the embryonic state. So you can term this comparative embryology. But it just reveals additional anatomical homologies that aren't visible in adult organisms. So here you can see we're comparing a chick embryo with a human embryo. And it says at some stage in their embryonic development, all vertebrates have a tail located posterior to the anus as well as a pharyngeal pouch. So all of us at some point had a tail, right? In our embryonic development, we had a tail. But chicks also have a tail during their embryonic stage. We also share a homologous structure in that we get these kind of pharyngeal pouches that grow in our what we would call our throat region. So these are just other examples of homologous structures. Now, we also get these things called vestigial structures or vestigial organs. And these are also some pretty interesting homologous structures. But basically, they're not usually used anymore, but they're remnants of structures that were utilized in our ancestors. So up here we can see an example of vestigial structures. So we have a salamander, and we can see that there's this bone kind of protruding off of the back of the pelvis that's not really used anymore. But if you compare that to a whale, you can see that that whale also has that same structure, as does a snake. So these are examples of structures that are no longer used, but they're still there. And they're there because of a common ancestor. Another interesting kind of concept to look at is this concept of biogeography. So it was Darwin's explanation for the geographic distribution of species. Closely related species tend to be found within similar geographic regions. But those in the same ecological niches in distant areas of either the country or of the world can be occupied by very different yet very similar looking species. So a great example of that is comparing a sugar glider with a flying squirrel. In the United States, we have flying squirrels. In Australia, we have sugar gliders. Well, there are two species that are pretty darn similar, but they've been separated based off of where they are geographically. So you can see that they've kind of evolved slightly differently, but they still maintain or they still have very, very similar characteristics. And that's what Darwin coined as biogeography.